This protocol is critically important because it standardizes an approach for the very difficult task of detecting motor-evoked potentials in the distal lower extremity muscles. The main advantage of this technique is that it consolidates the literature associated with lower extremity stimulation, which often focuses only on the tibialis anterior and uses a variety of techniques.
We have studied this technique in neurologically healthy individuals as well as those with stroke. However, the importance of the technique is in standardizing lower extremity assessments. The technique allows for the opportunity to identify the gaps in our understanding of motor control during gait and revisits research that did not allow for the control of interfering factors.
Demonstrating the procedure will be John Kindred, PhD, a postdoctoral scholar, and Brian Sentz, a research coordinator, both from our research laboratory.
Begin by uploading the subject's MRI files into a neuronavigation system. Then manually co-register the MRI to anterior and posterior commissures so that the Montreal Neurological Institute Atlas can be used. Next, reconstruct the skin and full curvilinear brain model by adjusting the bounding box around the skull and brain tissues. Identify 4 anatomical landmarks using the skin model, including the tip of the nose, nasion, and supratragic notch of the right and left ear. Now place a rectangular grid over the leg motor area in each hemisphere. Position the centered row of the grid at the center and over the gyrus of the leg motor cortical area. Then position the medial column of the grid parallel and adjacent to the medial wall of the